So I would like to thank Remedios and all the FACE network for this opportunity to present. So Remedios asked me to introduce the different topics that will be presented and discussed for these 2 days. So that's what I will try to do on my with my knowledge of a microbial ecologist. So this is the look of a microbial ecologist. So first, cheese microbiota. It can be considered as an evolving assembly of microbes, house microbes, and inoculated microbes. So as house microbes, we can consider the microbes coming from raw milk, which are under the influence of practices, farming practices, and also microorganisms coming from the cheese-making environment, such as the wood surface, brine. And all these microorganisms are, of course, essential for product typicity. But in addition to them, microorganisms also are voluntarily inoculated into milk, and they are composed of commercial starters, of autochthonous microorganisms as consortia, and also coming from traditional practices such as backsloping. And they are all important for the acidification and ripening process of cheese. So finally, traditional cheeses are composed of complex and specific microbiota, and they deserve better understanding of their diversity and their ecological drivers. So in the recent years, new methodologies were available for biologists based on DNA sequencing. And in this family of new methodologies, we use the one which is called metabarcoding, which is based on the sequencing of the DNA, a portion of the DNA, which allows us to identify the microorganisms present in milk and cheese. So using this methodology, we set up a project that we called in France MetaPDO-Cheese. And the objective of this project is to characterize the microbial diversity in milk and PDO cheeses, French PDO cheeses. So we set up a sampling design, and we managed to collect 2,400 cheese samples from core and surface of the cheese, and the 400 milks that were used to make this cheese. This cheese represented 44 different PDO cheeses, French PDO cheeses, from 7 different cheese technologies. And overall, the sampling, the total sample were composed of 51% of farmhouse productions and 86% of raw milk productions. So with all the data that we get, overall, we managed to identify the different species present in milk, cheese surface, and cheese core. And so as regard milk, we were able to identify more than 1,400 species in the 400 samples of milk. In the cheese core, we found close to 600 different species, and on the cheese surface, about 800 different species. Several of these species were shared between milk and cheese. Between cheese core and cheese surface, up to 400 species are shared, and between milk and cheeses, more than 100 species were shared. So if we have a closer look to the composition of these 2,400 cheese and milk samples, we compare them based on the composition of their bacteria. And we could find that the profiles of these samples were clustered depending on the origin of the sample. So on the right you have the milk sample in green, and on the left the cheese core samples and cheese surface samples. And if we have a closer look to the milk samples, then we can find that the species, the dairy species, was an important factor which contributed to define the milk composition as the bacteria in milk. Now, if we have a look to the data considering the technological cheese families that were studied, you can see that the samples were clustered according to the technological family. So we had 7 technological families. The first one is lactic cheese with blue bean rind, which comprised 14 different PDO cheeses. The second one was lactic cheeses with washed rind, which comprised 2 PDO cheeses, and so on. And so you can see from the groups of colors that the samples distributed in these 7 families were grouped according to these families. This was the samples from the cheese surface, and we did the same for the samples from the core of the cheese and found similar ordination of the samples. And if we look at the gross composition of these samples at the scale of the family, when we look at the composition of the microbiota, it is in agreement with the previous knowledge that was acquired using culture-dependent approaches regarding the major bacterial groups present in cheese. And we are awaiting for a complement in the data concerning yeasts and molds. And so we will also compare the composition of the yeast and molds in the different cheeses. So here we can have a closer look to one of the families, one of the cheese families, the uncooked pressed cheeses, which comprise 9 different PDOs. So we can see that there were major differences between the PDO cheeses. So from the left to the right, you have the 9 PDO cheeses and the different samples which were analyzed within each PDO. And we found differences in the proportion of bacterial species assigned to specific groups such as actinobacteria or proteobacteria. If we look more precisely to the data of one of these PDO cheeses, then we could show that the samples within this PDO were separated, and we could distinguish the samples from farm cheeses and those from artisan cheeses. This is only an example because we worked on a lot of different potential factors that could contribute to the composition of this microbiota. And overall, in the study, we identified different potential drivers of this microbiota, such as the dairy species, the PDO requirement, the production type, farmhouse or artisanal. We also found differences based on seasonal dependent factors, and many other factors are still under analysis. So, for the future, we could go a step further in these approaches using multi-omics approaches, because what I presented was focused on DNA analysis, but we can go further and use approaches based on the analysis of proteins and other metabolites, and then combining all these approaches, we could get a better insight into the microbial diversity at the strain level, between interactions between microbiota composition, microbiota functional potentialities, and cheese biochemical properties. With our objective, which is to enrich the scientific background for traditional cheeses' specificities. So now that we have evidence that there is such a high microbial diversity in cheese, what can we say about the relationship between microbes in cheese and human heirs? So ingestion of microbes through the diet, everyday diet, can represent up to 10 to the 6 or 10 to the 11 microorganisms per day. They are ingested with a solid matrix. It is a high biodiversity, and also pathogen can be present in these microbiota. So different potential impacts of cheese microbes on heirs were reported in the literature. Microbes can be beneficial through the production of metabolites, such as organic acids, vitamins, essential amino acids, specific fatty acids. The living microbes in this matrix can act as probiotics, and the microbes can also modify the cheese matrix via microbial enzymes and be associated with the production of bioactive peptides. They can improve the digestibility of the matrix. They can improve the availability of minerals. They can reduce metabolites with adverse effects, such as lactose or mycotoxins. And they can also reduce allergenicity. But also, they can cause toxin infections. So, question remains about the potential interactions between microbiota in raw milk cheese and interactions with the immune system, the human immune system, interaction with the gut microbiota, and the possible co-evolution of this microbiota over the millenary history of cheese production and consumption. So we cannot ignore the potential risk associated with the consumption of cheese and raw milk cheese. From the last report of the EFSA in 2017, it was shown that cheese represented 2.2% of total foodborne outbreaks reported in EU for the year 2017. And from a report in France by the DGL in France, pathogen responsible for outbreaks associated with raw milk cheese, the most frequent ones were Listeria monocytogenes, Shiga toxin-producing E. coli, and Salmonella. Okay, so I told you about potential interactions between microbes and the human gut microbiota. What do we know about that? Though only a few studies yet were set up to ask this question. They show that several populations present in cheese can survive during the transfer to the digestion process, maybe because they are protected by the cheese matrix, but usually they do not colonize the gut microbiota. But they can modulate, they can interact and modulate the gut microbiota after cheese consumption. And the responses that were evidenced were different depending on the cheese type. So at the moment, it's difficult to draw only unique response about the consumption of cheese and gut microbiota. We don't even know which are the microbial drivers associated with these responses. We think that lactic acid bacteria can have a role, of course, but what about other bacteria? What about yeasts? And is it needed to have simple species or microbial associations to have an effect? Raw milk, cheese can also modulate the immune system, and several epidemiological studies were set up to ask these questions, and they show that raw milk consumption would contribute to the protection of children against asthma, allergies, respiratory affections, and cheese consumption would also contribute to protect the child against atopic dermatitis and food allergies. We still don't know exactly what are the relations with the whey proteins or with the microbiota. So, to summarize, beneficial effects can be associated with cheese consumption based on its matrix, so the proteins, the lipids, the minerals, and the vitamins. The fermentation process can improve the composition in bioactive peptides and vitamins. And the microbiota in cheese, composed of living microorganisms, can potentially interact with the immune and digestive systems. Specifically regarding raw milk cheeses, the unprocessed raw material, the raw milk, can improve the preservation of proteins, lipids, and vitamins. And the microbial diversity is important, as you know, for the sensory richness of the product, and can also increase interactions with the immune and digestive system. Several drawbacks are also associated with cheese regarding the matrix with lipids and salt. And but there is recent work published which shows that the responses to the consumption of fat may be different when it is ingested with the cheese matrix or separately. So a lot of questions are still running. Of course, there are pathogens, potential pathogens, and several hypotheses are still studied regarding the protective effect against cardiovascular affections, the reducing blood pressure, antimicrobial or anti-inflammatory properties, or anti-oxidative activities. So now I would like to share with you what are the possibilities that we have to manage the balance between risk and benefits. We have to find a trade-off between safety, microbial diversity, and sensory richness. So one of the first approaches to manage risk and benefits is a targeted approach. It can be based on biopreservation. Biopreservation relies on microbial diversity in milk and cheese that is used as a barrier against pathogens. Mm-hmm. So isolates, microbial isolates, can be screened for their potential to produce antagonistic metabolites or to compete with pathogens, producing organic acids, decreasing the pH, producing bacteriocins or hydrogen peroxide, in order to inhibit the growth of pathogens and spoilers. And then we could improve the microbial safety and quality of fermented and also non-fermented foods, and preserve sensory and nutritional properties of the food. So several approaches can be used to design antimicrobial consortia, depending on the pathogen that you want to inhibit. In a work in our research group, we worked on Listeria monocytogenes, for example, and we started with raw milk cheese that was produced in a farm with very low prevalence of pathogens, very low prevalence of Listeria. And we isolated microbes from this cheese, and we were able to simplify the microbial community down to 4 strains that were still inhibitory against Listeria. But all these 4 strains needed to be together, they needed to cooperate to be able to inhibit Listeria, because individually the strains were not inhibitory. Now, if we look at another pathogen, Shiga toxin-producing E. coli, the case was different. Several individual strains were able to inhibit STEC, but they had synergistic effects in strains associations, and these associations were different depending on the cheese technology, which means that you have to design different consortia depending on the pathogen and depending on the cheese technology. Another alternative to biopreservation could be to consider managing the risk and the benefits by the management of microbial resources from the primary production environment across the cheese production chain. The objective would be to understand the drivers of cheese microbiota for safe and healthy raw milk cheese. We wonder what would be the influence of the overall farm management system, and several projects are running in a French network called RMT Terroir on these questions. So in our group, we also recently studied the microbial transfer from the dairy farm to the cheese. We studied the transfer, we compared the microorganisms present on the teat surface of the cows, in the milk, on cheese core and on cheese surface. And we could find that several microorganisms were shared between them. And in particular, 85% of the microorganisms present in milk were also present on the teat surface. And 27% of the microorganisms present in cheese, on the core and in the surface, were also present on the teat surface. So teat was considered as a potential source, a potential reservoir of diversity for the cheese. A research group in Italy even went further than that and showed that colonization of the human gut could be, microbes colonizing the human gut could come from bovine bacteria present in Parmesan cheese. So our objective is to go all the way down from the environment of the farm to the consumer. Mm-hmm. So we have to find the right balance between hygiene and excess of hygiene, because of course hygiene is mandatory. Hygiene is the principle and practices for health conservation, but due to the increased pressure due to the risk of pathogens, drastic hygiene practices along the production chain from farm to cheese are applied, and they are responsible for a lower diversity in the milk. In addition, intensification of production practices is also associated with major changes in the microbial composition of farm environment, milk, and cheese. As shown on this chart, the cheese variety and the microbial diversity decreases with the trend in industrialization of the production. Mm-hmm. 3 minutes. Okay. There is also a trend for an urbanization and highly transformed food, and this could be associated with an increase in autoimmune infections. So altogether, these factors raised issues, ecological and health issues, with a loss of microbial diversity and a lower exposition of human to foodborne and environmental microorganisms. And we wonder what would be the interactions with the gut microbiota and the development of autoimmune affections. So to conclude, I would like to share with you some thoughts about the possibilities to find solutions through systemic and transdisciplinary approaches. We should consider the whole chain from the primary environment, the processing, and the consumer with the public health considerations. We have to find the right balance between microbial inputs at the different stages of this chain and hygiene. We have to evaluate the impact of our practices on the microbiodiversity in natural and anthropogenic environments and the consequence on animal health and human health. And to be able to do that, we have to consider the role of the human beings inside in these questions, from the farmer, the cheesemaker, different stakeholders and consumer. We have to consider the way they think about the nature and the way they think about the microbes and the confidence they have in their own know-how. Because the way they will respond to the different pressures and the different advices they receive depends on that, and it will affect the feedback they give and the way we can find the right balance between microbial inputs and hygiene. So, of course, this will need very comprehensive approach, integrative and transdisciplinary. So thank you for your attention.